Okay, so so Neisseria and Haemophilus are kind of special because they're gram-negative, but they don't grow a McConkey. Okay. When you have in your chapters, so you have Neisseria species as one chapter, and then you have Haemophilus and the Hayseck group. So Haemophilus is in the Hayseck group. That is a whole nother chapter. So you're going to have Haemophilus to begin with, and then you're going to have a whole bunch of other kinds of organisms. And I believe I already told you that the Hayseck organisms are associated primarily with endocarditis. Okay, but Haemophilus, the ones I'm covering in this lecture, not so much. Okay. So, one thing I can tell you about these guys, they need CO2 to grow. So they are capnophiles. Some of them need some very special handling. So, common characteristics. Gram-negative, okay, capnophiles. Many are part of our normal flora. So, Neisseria gonorrhea should never be part of your normal flora, but all the other things could be. Okay, so there are other Neisserias out there that are part of your normal flora. One of them is one of the pathogens that we're looking for in Neisseria meningitidis. So that's normal in the oropharynx. It's crazy. But these things can be pathogenic in the upper respiratory tract. Some of them can be pathogenic in the reproductive tract. But they do have some very fastidious growth requirements between the two of them, like between the two groups, right? That's why we always include chocolate agar if we need to be looking out for Neisseria or Haemophilus. Okay, so basic stuff for Neisseria. These guys, Neisseria will look like two little circles next to each other. But when you look at them, they're not really circular. They look kind of ovally or like two little coccobacillus. If you could get like super close to these, what they actually look like are coffee beans or kidney beans that are with the little indent toward each other that makes that little gap in the middle. So, you will always see these things like in pairs as a predominant characteristic. So like you see this guy, oh, we got one single guy here, and we got one here, and we got one here, but then we got a pair, we got a pair, we got a pair, we got a pair, that's a pair, that's a pair, that's a pair, like they're paired up. Okay. A lot of times, especially if it's in something that's mucoid, so if it's in a sputum, urethral discharge, vaginal discharge, very mucousy, you will see them inside of white cells. So you'll see them inside of the neutrophils. So anytime that you see intracellular gram-negative diplococci, think Neisseria. So we do have a bunch of them that are normal flora, but primary pathogen, true pathogen is Neisseria gonorrhoeae, of course. Things that you do at the bench, you're going to do the catalase test and an oxidase test on them. Okay. Then you're going to do some identification testings, and once you get an identification on it, you also need to do a beta-lactamase test on it, because we need to know what kind of drugs to give. We now start treating Neisseria gonorrhoeae at the 3rd level of the cephalosporins, 3rd generation. We didn't used to. They used to be susceptible to penicillin. That doesn't happen anymore. They used to be susceptible to the 1st generation cephalosporins. That doesn't happen anymore. They're getting progressively resistant because it's been, oh, well, we'll just treat them with this. Oh, we'll just treat them with this. Oh, we'll just treat them with this. And now a few years ago, it was big in the media. Oh, we have resistant Neisseria gonorrhea. I'm like, we've had resistant gonorrhea for a long time. But they wanted to make the public aware so that, even though you've been treated, it might not have worked, that kind of thing. We'll talk about the collection techniques in a bit, okay, because I want to get to the individuals first, okay. So, here we go. Never ever use a cotton swab, because the fatty acids in the cotton fibers are toxic to some of the Neisseria. If it is something that could have Neisseria, particularly Neisseria gonorrhoeae, growing, do not refrigerate it. Keep it at room temperature until it gets plated. Or keep it warm, either way. Don't put it on freezing cold media, because cold kills it. It's a very persnickety little bug. Okay, so GC can be used 2 different ways. This could be genital culture, or it could mean gonococcus, which would mean that it's Neisseria gonorrhea. Okay, but this, in this case, means, is it a genital culture? And if it's a genital culture in the office, not all doctors do this, but some doctors still do it. They'll order the Gembeck plates, and they'll actually swab right after they collect it onto the media, so that we have a good chance of recovering it. They'll still send you the swab so that then you can put it on your blood and chocolate and Thayer Martin. But it's best right from the patient when it's nice and warm and in its home environment. Okay. And you'll notice that you roll a Z or you swab a Z on here. Okay. I told all of my techs to not swab across it this way, but to swab across it this way, so that you could tell where you struck it versus where it was swabbed. So you went in a completely opposite pattern from the Z. How you do it wherever you are depends on how they want you to do it. And if they don't tell you, you can form your own opinion. Okay. But you do have to, once they roll it on here or swab it on here, you still have to streak it out. Otherwise, you will not have isolated colonies. Okay. So what happens is this is in a little plastic case. And they just swab the Z on there. They clip the case shut, put it in the plastic bag with the little CO2 generator. See that little pad down there? And it comes to you. Sometimes you don't get the little CO2 generator because a lot of them don't know what this is for. But you get this, and it's still way better than not getting it. So typically, so you guys can, no, you can't. All right. I just want to make sure that what you were saying was exactly the same as what I was saying, because it's really huge for some reason. It won't go any smaller. All right, never mind. So we put it on blood and chocolate, and then you put it on Thayer Martin, or this is modified Thayer Martin. Okay. And the modified Thayer Martin is basically, it is a chocolate plate that has antibiotics added to it. So it has antibiotics to inhibit Proteus, inhibit the gram-positives, inhibit fungus, and like it's got 3 or 4 different antibiotics added to it, so that we're really only trying for the Neisseria gonorrhoeae, but other Neisserias will grow on this as well. Okay. Most cultures are held for 48 hours. Okay. Sometimes if you start seeing little tiny growth at 48 hours, you might want to keep it that extra day just to make sure that it isn't something, because some of them grow a little bit slower than others. So it is what it is. So, is it Neisseria or isn't it Neisseria? Well, typically the Neisserias will grow, all of them will grow on chocolate. Okay. And Neisseria meningitidis and Neisseria gonorrhoeae will grow on Thayer Martin. Did it grow on your sheep's blood auger, your blood auger plate? Here's where we come into things happen different in Mrs. Murray's lab. All of your Neisseria are supposed to grow on blood auger except for Neisseria gonorrhoeae. Unless you're in Mrs. Murray's lab. Because I had a problem with my son Neisseria grow on blood agar. So I went and got a whole nother kind of Neisseria gonorrhea, right? It also grows on blood agar in my lab. I don't know why, but when it's coming from a patient, it shouldn't be growing on the blood, but you'll see it growing on the chocolate and the Thayer-Martin. And no, I am not going to inoculate somebody's genital region just to get a good specimen for you guys for your practical. These are going to be homegrown here, like with what I got, so it might grow on blood agar for your practical. Okay? But, so you'll see gram-negative diplococci. A lot of times they'll be intracellular, so you want to be looking to see if they are intracellular. Okay. And you're going to do a catalase, which should come up positive, and it comes up very positive. It's not slow in any way, shape, or form. Your oxidase will come up positive. And then typically what we do is we do enzyme testing to determine what species it is. Okay, and we'll learn about that a little bit further down. Okay, so we have different ways that we can do our enzyme testing. We can do the Bacti-cards, or we can do the Neisseria enzyme testing that's liquid in the things. And then once we figure out who it is, you need to do that beta-lactamase testing to find out if they're resistant, if they produce beta-lactamase that would make them resistant to some treatments. Okay. So, Neisseria lactamica is not typically pathogenic, okay, but it is a Neisseria, and you may run into it, okay. It will grow on blood and chocolate. Okay. But some of these tend to be identified as meningitis instead of lactamica because they're late lactose fermenters. So what we can do for that is we can also do something called these are all capital letters, ONPG. Okay, capital O, capital N, capital P, capital G. And we do that to differentiate between Neisseria meningitidis and Neisseria lactamica at 24 hours, because it could be a slow lactose fermenter. Okay. Now, this is one of the reasons why we don't usually do the sugar testing. Instead, we do the enzyme testing to find out what enzyme the actual organism produces and has. Okay. So, there is a very slim chance that you will ever diagnose somebody with Neisseria lactamica. Do you understand that? Because they're typically non-pathogens. And in my 14 years of reading microbiology, I never had a Neisseria lactamica as a pathogen. So, the enzyme that Neisseria lactamica has that the others don't have is it has the beta galactosidase. Because if you remember, you have galactose and glucose that make up lactose. So it has to be able to use the galactose. So in our Neisseria enzyme testing kit, the one with the liquids, it'll turn a pretty blue color. Okay. Okay, so we don't do this typically. If you end up doing a 4-hour identification test, like a little tilt test, then that will have the carbohydrates in there. But we don't do that frequently. We typically check for the enzyme, see what's there. Okay, so this picture represents one of multiple types of Neisseria enzyme tests. If you look at this setup, okay, there's liquid in the bottom there. This is a plastic tube, and it has a white plastic cap and then a red plastic cap on top of it. Okay, so what you do is you take both of those caps off at once. They come as a unit. Okay, you put your critter in there and you incubate it. After you incubate it, you look to see if it changes color. If it changed yellow, then it could be Neisseria meningitidis, mucosa, or subflava. Okay. If it turns blue, it should be Neisseria lactamica. That is with just incubation. Okay. So then, if it didn't turn colors, what you do is you take both caps off, and you take the red cap out of the white cap, and you put the red cap back on, and invert it because there's pro reagent on there. Invert it a couple times. I think it gets put back into the incubator. I can't remember. It's been so long since I used this one. But it will turn bright red if it's Neisseria gonorrhoeae. Okay. Or it'll turn like it'll be colorless. It'll be a really super pale yellow or even a peachy color. If that's a B, that's wrong. It's M. cataralis now. Okay. So notice that there's all these other things here too. Okay. King Allen denitrificans is typically not found in the same places where we would find Neisseria gonorrhoeae. Okay. So it's not typically a pathogen in the same locations. So look at the location. Where did this come from? And all of these things should grow on blood agar, and Neisseria gonorrhoeae shouldn't, but haha, this is Mrs. Murray's lab. So notice that, subflava could be yellow with just incubation, or it could turn red, but you'll be checking that, whether it's growing on selective media or not. Okay. So these things are very easy to use, but they're very expensive. So we don't use this exact one, we use a different one. Okay, so Neisseria meningitidis. Neisseria meningitidis could be part of your normal flora. Okay, but if it gets into the cerebral into the cranium. If it gets into the cranium, gets into the bloodstream, your patient could be dead in a day. Okay, so you want to be very aware that if it's, in where all the normal flora is, okay, but if it's even a tiny bit beyond your normal flora, you definitely need to work this up. Okay, so we see this very frequently in cerebrospinal fluid, of course, when we have bacterial meningitis. Neisseria meningitidis is called meningococcus. Okay, like the Neisseria gonorrhea is gonococcus, this is meningococcus. Okay. So meningitis can hit hard, turn into meningococcemia very quickly, which can turn into Waterhouse-Friderichsen syndrome and kill you. This can all happen very quickly. Like, super quick. Day or two, done. Okay. So, this is why we have meningococcal vaccines. So, it is encapsulated. It is a gram-negative diplococci. It's going to be catalase positive. It's going to be oxidase positive. It'll grow on the blood. It'll grow on the chocolate. It'll grow on Thayer Martin. Not that you're usually looking for meningitis in any genital cultures or anything like that. But it will grow on there within 24 hours. It grows very well. It only can ferment glucose and maltose, not lactose. And the enzyme that it uses is the gamma glutamyl peptidase, which will turn a yellow color upon incubation, that first incubation. It'll be yellow afterwards. See? Neisseria meningitidis. You incubate it, turns yellow. Okay. So, we can do some serology testing. I'm looking to see if I still have it on here. It's on the other PowerPoint. Okay. Serology testing. So, for cerebrospinal fluids, we can do rapid antigen testings for cerebrospinal fluid to find out if it's one of the 6 most common bacteria that can cause meningitis. Now we do CSF panels on the Cepheid IDs. So, that's a good thing. Because you had to boil this cerebrospinal fluid to get the capsules going. Alright. So, yes, it is oxidase positive. Yes, there are still people out there that put oxidase on their cultures. I'm like, why? You guys know we have multiple ways of doing our oxidases, and we even have the little oxy strips, which, yes, Eddie, you do have to add a little tiny bit of water to. You said, don't you have to add water to them? I'm like, I don't know. I didn't read the thing yet. Just do it the way it is. Yeah, I know. So, well, it does. The strips you have to. The windows you don't. It's good stuff. But glucose, maltose, and lactose, not there, right? So, again, this is where it turns out. 24 hours, they look, medium, kind of like creamy. They kind of have like a pinkish, grayish tinge to them. So it's like, they're like gray, tan, pink looking. That's 24 hours. 48 hours, very flat, gray colonies. So they look very different from day to day. But you will get to see how different they look. Alright, so this is the bad guy. Bacterial meningitis can cause bacterial meningitis. Anytime that they have somebody who they suspect is having meningitis, everybody that's in the ER with them and in their presence is supposed to have a mask on. Can't tell you how many times they're like, oh, we think you might have meningitis. I have a sticking out, exposed, no mask, nothing. So it's okay. Young adults, okay, teens to late 20s, high point. Like, they're the ones that get bacterial meningitis a lot. Why? We don't know. Okay. College students, this is why we now make sure that we have meningococcal vaccinations done. People that are going into the military, yes, you all need your vaccinations. Okay. So the organisms get in the nasopharynx, and how many teenagers want to run around with tissues when they're sick at school? They don't, right? So they do this. Right? Like, use the tissue. Because the more that you're sniffing up, you can force it across there. And they're pretty good at getting there. So what happens is they'll get a really bad headache. Then their neck may get stiff. They may just not want to get out of bed. They may have their head under the covers because they get sensitive to the light. Okay? Light sensitivity is a sign of meningitis. So that's a really, really bad thing. It's also a sign of migraine, so, it's kind of hard to tell the difference between the two of them. Unless, of course, you're like, move your head. Do this with your neck. Does it hurt? Okay, good. So, transmitted by aerosols. So the big thing is that we can have asymptomatic carriers because it can be part of your normal flora. Okay, I have a DVD here that if we get a chance, I would love for you guys to see this, because it talks about, it shows you how fast you can die from this. It's a really awesome movie. Okay. Meningococcemia, so it can go to the cerebrospinal fluid, it can get into the bloodstream. In the bloodstream, it causes, it's almost like DIC, causes little thromboses in your capillaries, and you'll get a little petechial rash, and then all of a sudden it goes to DIC. And with the DIC, then you can actually get internal bleeding in the organs, and then the organs will shut down and you die. So, little petechia, right? These aren't bright red, but they start out as red and then they get darker as they go. And then they can spread and get worse and worse. Okay. This, what organ is this? Do you guys know? Kidney? That's a heart. That's a heart that has been taken out. It's got clot everywhere. Yeah. So, it's terrible. Okay. So, typically, first line we go with penicillins. So, we've kind of moved away from the penicillins. We go with macrolides more commonly now, or we go into a third generation, fourth generation, like some sort of cephalosporin, but chloramphenicol works really well. And if somebody has a rash, they've had a headache and they start getting a rash, there is a really easy way to determine if it's an actual rash or petechia. Does anybody know this? Take a glass and push it on their arm, on the rash. If it goes away, it's a rash. If it turns white, if it stays pink or red, it's petechia. So you can take the glass and roll it across it and check and see if it's disappearing or not. It's very helpful if you ever want to have children and you want to check.
I got a vaccine.
Yeah, well, I got all of their vaccines. I got all Russell's vaccines, but you know what? He had a really bad headache one time, and I'm like, stick out your arm. You have a rash anywhere? Oh, I don't think so. I'm like, move your head around. He's like, go away. So, Neisseria gonorrhea is known as gonococcus. Okay, so that is where you may see GC sometimes. So remember that GC could be genital culture or gonococcus. Okay, a lot of times too, instead of putting GC down, which is our shorthand in micro, sometimes instead we'll just put N-gon, or a big N with a little G, which is not no growth, because no growth is capital N, capital G. Okay, so that we can do a real fast thing. Now everything is documented on the computer, so it makes it a lot easier for you that you can actually type in as much as you want. All right, but we used to have a space about this big to write our notes for the day. So everything had to be shorthanded so everybody knew what you were talking about. Microscopic, very common. Intracellular, gram-negative diplococci, very, very common to see them as intracellular. Okay, you're going to see a ton of your neutrophils in there. Okay. This will be on your gram stain from the specimen. Now, do we normally do cervical and vaginal gram stains? Depends. So, a lot of times you might do them on patients coming into the ER, or specifically from like the gynecology clinics or offices or stuff, because they know that the patient they know the patient history more, or they don't know the patient history, and it could be gonorrhea, and we want to nip it in the bud and get them treated and start that whole protocol. Okay, so typically you don't see any growth on blood, except for here, but you'll see growth on chocolate. And again, look, this kind of looks like that Neisseria meningitidis. That's a 24-hour culture. Okay, and then when you get it on Thermartin, they look the same. And they both are catalase positive, and they're both oxidase positive, and they look the same on the microscope. It's like you don't know until you do that enzyme testing. Okay, so it's very important to get the enzyme testing correct. All right. Now, you could do a nitrite test on it, but how long does it take to do a nitrate test? Do you guys remember the nitrate test with the zinc? It was in a broth, right? And then you had to incubate it at least 24 hours, sometimes up to 48 hours. Like, that's not going to help you get an ID out fast, right? We don't worry about that. So you're going to do your beta-lactamase testing, and you're going to do your enzyme testing, okay? And what we're looking for is this prolyl aminopeptidase. And the book has another longer version of the same thing. I can't remember what it is. Prolamino gamma. I don't know. I don't remember. So, but as long as you realize that prolopeptidase is pro, which is, pros get gonorrhea. That's how I always remember that one. Turns red. Okay, so look at the source. Where did this come from? If it comes up positive for that pro and it's not somewhere where you would suspect Neisseria gonorrhea, maybe it's not. Maybe it's some other Neisseria that will grow on blood. Go check your blood. Maybe it's Kingella. Kingella is usually a deeper infection than something on a mucosal membrane. So the other thing is they won't all grow on the Thayer Martin. So did you take it from the Thayer Martin? That's the other thing. Testing for gonococcus should come from the Thayer Martin. Not the catalase and the oxidase, but the other stuff. When you're doing enzyme testing, take it from the Thayer Martin. Because that's your best chance that that's really N-gon or N-min. So when you get that pro-positive, prolamminopeptidase positive, you can put it out as presumptive Neisseria gonorrhea. Now, what do we do instead typically? PCR. So we can do a PCR swab, immediate, like you'll get it the same day, good to go. So that's not too bad. Okay. All right. Here is another gram-negative diplococci that is part of your normal flora, but can be an opportunistic pathogen if it gets into weird places or overgrows. So, Moraxella cataralis is catalase positive, oxidase positive. It's a gram-negative diplococci. Moraxella cataralis tends to not be intracellular, but it could be. It's just not going to be chock full of them. Okay. This will grow on blood and chocolate. It's not supposed to grow on Thayer Martin, but it could. It just depends on the strain that you have. Some people have been on lots and lots of antibiotics over the years, so it could actually grow for them. Moraxella cataralis tends to, when you go to pick it up from the colony, it'll not be smooth consistency and creamy like the other Neisserias. This one actually, they like to adhere to each other. You can actually, when you can take a loop and push the colonies around on the plate. That's so they call them hockey puck colonies, because you can push it around with your thing. And I will show you guys that so that you understand. Now, does that mean that the whole colony is? Maybe not. Some of it might break off. You might be able to push part of it, and but they tend to not be smooth and creamy like the other guys. You will see that. I'm making you guys do the gram stains on all these things because you need to see what the consistency difference is with all these. The staphs and streps, the consistency was all the same. But these guys, they have a little different consistencies. Okay. And the Moraxella cataralis didn't have any fermentation. So it doesn't have the NET enzymes that'll do this. So it'll be a colorless light yellow. One of the things that we come up with commonly here, because the pro reagent that you add is pink, okay, it looks like a really light peach color. Okay, so the peach would be this. On the Bacti cards for the Neisseria testing, they actually have a space for the butyrate esterase test. So if the butyrate esterase test comes up positive, it's Moraxella cataralis. Okay, so that's nice that they have all 4 of the enzymes that you're looking for. So you can tell between the 4 most common things that you'll identify. But the NET test that is in the tube test, not so much. So that's the one that's going to be after you add your pro. It's going to be colorless or a peachy color, light yellow, something like that. Not bright yellow, light yellow. Okay. So, how do we tell all the differences between these? I will flat out tell you that going down the line from top to bottom, what I did was Lactamica has lactose as well as glucose and maltose. Meningitis has maltose as well as glucose. Gonorrhea has only glucose, and then cataralis doesn't have any of them. Okay, these are the sugars that they will ferment. You need to know that. You absolutely need to know that. Why? Because it is extremely important to the identification of these things and the BOC. So if you don't know that and you only know the enzymes, you're not going to get the questions right on the BOC. Okay, and I believe I even have them have questions on the exam that have sugars on them. So, okay, so the enzyme tube, you can do 3 enzymes in one tube. So beta-galactosidase would be lactamica. It'll turn blue upon incubation. The gamma-glutamylaminopeptidase for meningitis will turn yellow upon incubation. After you add pro, there you go, there's the other one, hydroxyprolamminopeptidase. Okay, after you add pro, that's gonorrhea. See, I color-coded the whole thing for you. Isn't that pretty? Because it turns red, this will be yellow. Okay, so these are the Bacti cards. Okay, these are the colors that they turn. Okay, so beta-galactosidase for lactamica, gamma-glutamylaminopeptidase for the meningitis, hydroxyprolol for gonorrhea, and then butyrate esterase, because I didn't want to have another B in the front, is for the Moraxella cataralis. Okay, so you can, if you want, you can put the number, the organism next to all these. Figure out how to make that happen. So, then I did a little who am I? Okay, so glucose, but not maltose, and no galactose. So who is this? Gonorrhea. So this one did no glucose. No, you did glucose, maltose, and lactose. So that's lactamica, right? And this one did glucose and maltose. And glucose and maltose would be meningitis. So, yes.
What does how do you say the M1?
Moraxella.
What does it cause? Like where?
So it causes ear infections, eye infections. It can cause pneumonia, which it has it up here. So otitis media for middle ear infections, conjunctivitis for eye infections, any upper respiratory tract infections. It can, not common for like coughs, colds, that kind of upper respiratory, but it can get into like, it could cause a gentle sinus infection, and pneumonia. And the reason why I say gentle sinus infection is because it doesn't really like to be outside of oxygenated areas, so it won't make it like jam-packed and cause all that pressure and stuff like you normally get. And so it'll be like, I feel really stuffed up, but I don't have excessive amounts of pain, and like it's weird. It's a weird sinus infection. Okay. So then we're going to do Haemophilus, okay? But we're not going to cover Haemophilus until next week, okay? So I want you guys to read the chapter on Neisseria and go through it. It pretty much will sum up what I've said, and then we'll start on the Haemophilus and the Hasek groups, okay?